Hello, this is a presentation for Unit 17, Human Immunity by Kian Mai. These are the topics I will be covering on this assignment. The first part of the assignment will be covering Section 1, showing an understanding of non-specific defenders against various diseases. An infection occurs when microorganisms invade the body, typically through the mouth, nose, or through cuts in the skin. Bacteria are single-celled organisms which can cause diseases such as meningitis and pneumonia. Skin diseases, for example, athlete's foot, are caused by fungi, and parasitic diseases such as scabies and malaria are caused by parasites. Infections are transmitted through direct contact, indirect contact, insect bites, as well as food contamination. Indirect spreading of infection occurs when a person is in close contact with a contaminated object, for example, when eating food contaminated with a bacteria or virus. The body has three lines of defense to protect our cells and vital organs from infection. The innate non-specific defenses are mechanisms which function to prevent pathogens from entering the body, as well as providing functions to eliminate invaders within the body. The skin is the largest organ in the body and acts as our most prominent physical barrier of defense. The epidermis of the skin provides a waterproof covering which prevents penetration of microbial organisms, chemical irritants, and toxins. Should the skin sustain injuries, a scab will slowly form a temporary barrier, while the dermis and the subcutaneous layer of the skin underneath heals. The body is also able to fight microorganisms and viruses through the secretion of various enzymes from the internal mucous membranes. These membranes are responsible for filtering infectious agents, pathogens, and allergens, whilst allowing nutrients to pass through. Stomach acid, pancreatic enzymes, bile can all be found in the internal gastrointestinal tract. Chyme movements throughout the digestive systems via peristaltic contraction also helps to remove microorganisms. Our nasal cavities are coated with sticky mucous membranes, which traps the microorganisms before they are able to enter the body. Another area prone to infection is the urethra tract. Female urinary tract is shorter in comparison with adult males due to genetic differences, which can provide a passage for external bacteria to enter the bladder. However, frequent bowel movements and bladder helps the body in flushing out bacteria for both sexes. When the body experiences an injury or infection, an inflammatory response will be produced as part of the immune system. Some of the responses include dilation of blood vessels forming gaps in the cell wall, allowing large blood cells to follow through, causing inflammation as a result. Large supply of protein will also flow through the localized area in which the infection took place, fueling the immune response, stopping the damage from spreading as well as promote healing. The area in which the infection took place will therefore appear swollen and warm to the touch due to the increased inflammation. Increased vascularization are also associated with an increase of phagocytes at the site of injury. This can enhance the inflammatory response because phagocytes may release pro-inflammatory chemicals when they're activated by cellular distress signals released from damaged cells. As shown in this diagram, when the skin is damaged, chemical signals will be released to alert the body to produce phagocytes, which are immune cells derived from macrophages, to move into the area, absorbing all the bacteria into a vesicle called a phagosome. This whole process can be understood as phagocytosis, which will be further explained in the next slide. Phagocytosis is a cellular process for eliminating particles, including microorganisms, pathogens, and foreign substances. The process of phagocytosis involves several phases, from the detection of the particle to be ingested, activation of the internalization process, formation of a specialized vacuole called phagosome, and maturation of phagosome to transform it into a phagolysosome. When the phagosome fuses with lysosome, a phagolysosome will be created, which helps to end the process of phagocytosis. Macrophages developed from phagocytes provide additional microbial protection by blocking as well as destroying pathogens. The next section of the assignment will show an understanding of specific immunity. Cytokines are complementary proteins which are crucial in regulating the immune system as well as inflammatory responses. There are more than 30 complement cytokines in total, which can be activated by the presence of pathogens or microorganisms. Cytokines interact with each other to produce responses, including the formation of phagocytes to remove unwanted particles, generating proteins to attack invading organisms, as well as initiating the inflammation process. Antigens are molecules found within the pathogen, which helps it to enter the body. Antigens cause the immune system within the body by communicating with the system to produce antibodies by binding with the receptors on the surface of the lymphocytes. Neutrophils are the most abundant white blood cells and are often the first to reach the site of infection. These medium-sized cells attach to the walls of blood vessels and block germs from accessing the bloodstream. Monocytes are the largest white blood cells and would become macrophages to carry out the phagocytosis process. Eosinophils target larger cells such as worms, infestations, and moderate allergies. Basophils are the rarest of the white blood cells, which secrete antibodies to act against the hypersensitivity reaction. Lymphocytes are small cells which has a large nucleus located within a small amount of cytoplasm. As mentioned in the previous slide, lymphocytes are a type of white blood cell which can be found in lymphatic tissues around the body. There are two types of lymphocytes, T cells and B cells. T cells mature in the thymus gland and function to eliminate particles, including pathogens and microorganisms. Helper T cells, the most common type of T cell, regulate the majority of the functions of the immune systems through the release of lymphokines that stimulate other T cells to attack foreign bodies. B cells mature in the bone marrow and produce immunoglobulins, otherwise known as antibodies, when activated. Plasma cells are a type of B cell which secrete antibodies into the system, whereas memory B cell helps to store the memory of various antigens in the lymph nodes to be used again if required. Immunoglobulin are glycoprotein molecules produced by the white blood cells, and they are essential for the immune system to fight against infectious agents, including viruses and bacteria. As can be shown in the diagram, they are chains shaped similar to the letter Y, with the smaller chains having the ability to bind to antigens. The common variations of the immunoglobulin are IgM, which consists of 5 regular immunoglobulins and will be the first immunoglobulin to present during an immune response. IgE is the largest immunoglobulin. There's generally lower levels of the IgE in healthy individuals, and the levels will increase with parasitic infections and allergic reactions. IgG is the most common immunoglobulin due to it being the primary antibody being transferred from mother to baby via the umbilical cord and placenta, providing additional protection. IgD is the least common antibody, primarily exists to become the antigen receptor to B cells. Self-tolerance is the ability of the immune system to recognize self-produced antigens as a non-threat while appropriately deploying a response to foreign substances. The balance of immunological defense and self-tolerance is critical to normal physiological function and overall health. Foreign antigens can be found on pathogens entering the body. They will then be remembered by B cells, in which additional protective mechanisms will be deployed. The majority of nucleated cells have unique surface molecules with highly concentrated major histocompatibility complex, also known as MHC. The immune system will therefore not target cells with MHC1 identification. On the contrary, MHC2 molecules are presented mostly in antigen-presenting cells, such as lymphocytes, macrophages, and dendritic cells. The main difference that these two molecules shared in terms of appearance is that MHC1 comprised of a single A protein chain with a smaller B2 microglobulin protein, whereas MHC2 consisted of 2 protein chains, an A and B protein chain, that are similar in length. Hypersensitivity is an immune response produced by the immune system due to failures of self-tolerance, for example, when the body is repeatedly exposed to the same antigen. The reactions of hypersensitivity can be considered undesirable, for example, hay fever, asthma, or eczema. Another example of hypersensitivity include autoimmunity diseases, which is when the body mistakes its own native cells for foreign elements due to an unknown trigger, attacking its own native cells in the process. The four primary types of hypersensitivity reactions can be displayed in this figure here. Autoimmune diseases can cause muscle pains, low fever, fatigue, and inflammation. Rheumatoid arthritis is one of the more common autoimmune disease, described by the NHS as a long-term condition that causes pain, swelling, and stiffness in the joints. Unfortunately, there is no known cure for rheumatoid arthritis. However, symptoms are treated with medicine such as over-the-counter drugs, for example, ibuprofen, or prescribed drugs. Physiotherapy also helps to maintain the mobility of the person living with such disease. Women and smokers are said to have higher risk of developing rheumatoid arthritis, as well as people who has got family living with such disease. Another common autoimmune disease is multiple sclerosis, or MS, which is defined as a condition that can affect the central nervous system caused by an abnormal response by the body, causing a wide range of potential symptoms relating to vision, arm and leg movement, as well as sensation. There are two primary types of MS, primary progressive and relapsing remitting, with the latter being the more common type. People living with relapsing remitting MS will gradually experience the worsening of symptoms for a few days or months before slowly improving over the same period. Treatments for MS include prescribing steroid medicines to ease the painful symptoms, as well as recommendations from a physiotherapist to reduce muscle stiffness according to MS Society. Organ transplant is a life-saving procedure carried out to replace any malfunctions of a person's organs with the donations made by the donor. Unfortunately, the immune system does not always accept the donated organ, viewing them as a foreign agent. The process which follows afterwards is called the rejection of transplanted organs. As discussed in previous section of the presentation, the immune system is normally able to recognize antigens on the surface of cells as self or non-self. In the case of a rejection of an organ transplant, the immune system may not be able to recognize the antigens presented in the organs, which may prompt it to initiate an attack. To improve the chances of a successful organ transplant, physicians will emulate the organs and tissues of the donor to match as closely to the recipient as possible. Medicines containing monoclonal antibodies to fight off foreign T cells will also be able to prove helpful during and after surgical procedures. The three main types of organ rejection will be shown in this figure here. Acute rejection is mostly experienced by recipients after the surgical procedure. Hyperacute rejection occurs when the wrong blood type is given, and chronic rejection occurs after periods of years or months when the immune system slowly damages the organs of the donor. The final section of the presentation will show an understanding of the biology of vaccination and concept of immunity. Active immunity refers to the production of antibodies within the body to resist a particular disease, either actively or naturally. Natural active immunity directly exposes the body to the infectious pathogens of a disease, for example, common cold. Once the body has been exposed to a strain of common cold, antibodies will be produced as part of the immune system, which will then be stored by memory B cells as immunity to that particular strain. Artificial active immunity is gained after the introduction of infectious pathogens into the body through the introduction of a vaccine. The immunity process is similar in comparison with natural active response. However, should the body become infected once the vaccination has taken place, memory cells containing suitable antibodies will bind to the infectious agents, destroying them. This is known as immunological memory. Natural passive immunity involves the transfer of antibodies between a mother and child at birth or through breastfeeding. Antibodies will be transferred during pregnancy through the form of immunoglobulin G, which can provide additional protection for up to 6 months. Artificial passive immunity involves acquiring protection through transfusion of monoclonal antibodies. Examples of this method include blood transfusion or injection. A vaccine contains a small amount of pathogens in the disease which aims at stimulating the production of antibodies within the immune system against the specified disease. Vaccines can usually contain specifically treated live cells, harmless fragments of a pathogen, as well as toxins produced by the pathogens. According to the NHS, benefits of vaccines include providing additional protection to the community, stopping the diseases from spreading to other people who cannot have the vaccines. The immune system also fights off the original disease much quicker. Since the launch of the UK government's booster program, protection against COVID-19 symptoms was at 90% for individuals receiving the third dose of vaccine. The immediate response of the immune system, which occurs after a pathogen enters the body, is known as the primary response. Time will then be required for the B lymphocytes to recognize the antigens. During this process, symptoms will begin to take hold of the body. As a result of the secondary response, should the body encounter the same pathogens again, antibodies will multiply quicker thanks to the memory cells fighting off the diseases. Herd immunity is the understanding when a wide number of people within a community is vaccinated, those who has the disease or unable to take the vaccine will be protected against the disease, making it harder for the disease to pass through to everyone, reducing the spread of infectious pathogens as a result. One of the main problems facing herd immunity is during the process of vaccination, some people may get infected before the vaccine has had time to work. Furthermore, pathogens such as HIV-1 or influenza may mutate very quickly, changing the pathogens it presented, causing more difficulties for the immune system to protect against these pathogens. Transmissibility and severity of the disease also affect the threshold of the vaccination rates for the community to achieve immunization. Herd immunity has become increasingly difficult due to our world becoming more connected, making the population become more susceptible to catching diseases, particularly proven during the COVID-19 pandemic. Measles is a highly infectious disease spread by airborne transmission. Individuals infected with the disease will show symptoms of fever, rash, heavy coughs, which could be fatal. As indicated in this chart here, the recorded cases of measles for England and Wales decreased significantly since the introduction of mass vaccination in 1968. A notification of measles fell progressively in between 1940 and 2012. A second dose of vaccine, which aimed at limiting the spread of measles within a wider community, was introduced in 1988, causing substantial declines of recorded measles cases. An article published by the Department of Health and Social Care in England in 2019 suggested during the late 1990s and early 2000s the coverage of vaccine for children of 2 years old dropped to below 80% due to the widespread link existed associated with autism. In 2013, there were increasing number of measles cases, and this has been attributed to the proportion of 10 to 16-year-olds who were unvaccinated. The fall in MMR coverage has led to potential for large outbreaks of measles to occur, especially in big cities such as London. In 2016, however, the UK has achieved sufficient measles elimination, with 95% being the vaccination coverage for school children aged 5 years. In 1979, the WHO declared the smallpox epidemic has been eradicated thanks to a worldwide vaccination program, and ever since 2002, there were no recorded cases of polio. An example of a vaccination program on a global scale was in the SARS-CoV-2 vaccine, which commenced in the United Kingdom in December 2020. At the moment, there has been over 100 million doses given in the United Kingdom and more than 7 billion worldwide, with a global vaccination rate being 42%. To increase vaccine uptake, the NHS has authorized pharmacists to administer vaccines. A recent article has suggested the lack of trust from ethnic minority groups due to systemic racism, lack of information presented from the local council with regards to vaccination uptake. Social norms could also influence people's motives. For example, those living in communities where it is a norm to take the vaccine will become more motivated to do so. This is the end of my presentation. Thank you for listening.